Hi guys, here's video number 3, and I'm gonna do it this way without me in it so that I can see what you're seeing easier. All right, so we did this lab on cellular respiration. So cellular respiration is taking that glucose molecule that was made from photosynthesis, that's sugar, and basically we used the same kind of sugar, similar, in this lab. And we used yeast, which is a single-celled organism that is living. So if it's a living organism, it needs energy. So it will take the sugar, the yeast takes the sugar and uses that as its food source. Now, when you burn sugar, you release energy, and you also release carbon dioxide gas. So when we look at our 3 flasks here, each flask had 50 milliliters of water, distilled water. Each flask, 1, 2, and 3, had 2 grams of yeast. Flask number 1 had 2 grams of sugar. Flask number 2 had 2 grams of sugar, but flask number 2 I boiled for 2 minutes. Flask number 3 had no sugar, just the yeast. All right, so did flask number 1 do cellular respiration? Did it produce a gas? Well, it looks like it did. Did flask number 2 do cellular respiration? Did it produce a gas? Nope. Flask number 2, the boiling must have done something to that yeast. What do you think it did to the yeast? Is the yeast still alive? It's got food. It's got everything it needs to do cellular respiration, and it didn't. So is the yeast still alive? Flask number 3, the yeast has no food. If you look at flask number 2, the balloon is almost sucked into the flask, and any air that was in there to begin with is sucked out. Flask number 3 has a little bit of gas in the balloon. It's not sucked in, but it didn't produce a lot of gas. Not a lot, just maybe a little. So, did we see yeast doing cellular respiration? I'm actually going to do a 4th video here. I'm going to take these flasks. I'm going to take this flask, and I guess I'll take one of these. And I'm going to put some methylene blue in. Okay, that'll be video 4. But in the meantime, I also did the chromatography. Remember, I took the piece of spinach and I rubbed it here on this filter paper, and then I dipped this whole piece of filter paper into alcohol, rubbing alcohol, and the rubbing alcohol went up it soaked up the filter paper, and as it passed through the chlorophyll right here, the pigments that make up the chlorophyll they dissolved in the alcohol, and so they kind of were taken by the alcohol up the filter paper. Okay, now I see right here, it's really hard, it's really light, it's hard to see. Right there, it's bluish green. Okay, so that is a pigment that makes up chlorophyll. And then up higher, here's pure yellow. There's another pigment that makes up the chlorophyll. Okay, so the bluish, the the yellow didn't separate completely here. Otherwise, this would be totally blue. So we would have blue, yellow. Blue is a heavier pigment, heavier chemical compound than the yellow. So the yellow could travel further with the alcohol. The alcohol ended way up here. There's nothing up here. So this just shows you the different pigments that are found in chlorophyll. And then the last thing we did today, I took some spinach and put it in some hot water. This was alcohol in this flask, in this beaker. And I was able to pull the chlorophyll right out of the leaves. So there's the chlorophyll. Okay, so there's video number 3 with our lab results.